DNA, the instruction manual for all living organisms.
Our DNA gives instructions to make proteins and molecules necessary for our well-being.
But sometimes our DNA mutates, and those mutations can turn into genetic diseases.
Millions suffer from incurable genetic diseases, but what if our own genes were the cure? With gene editing, scientists can bioengineer a tool to edit living cells and eliminate the disease-causing mutation. Doing this would reprogram the cells to be healthy.
If we compare gene editing to fixing a damaged house, you need specific tools to fix specific damage. Just like you need to figure out what tools are best to cut this piece of wood, you need to figure out what techniques are best to cut DNA.
ZFNs were the first widely used programmable nucleases, improving success rate of gene editing by 10%. Another technique, TALENs, while similar in procedure, is easier to use. But both these techniques take a lot of time and money to program, which is where CRISPR revolutionized the field. It is composed of two parts, a Cas9 nuclease and a guide RNA. The gRNA acts as a blueprint for where to cut, and Cas9 acts as the molecular scissors, creating a double-stranded break. DSBs can then either be repaired by non-homologous end joining, allowing the DNA to create random mutations to repair itself, knocking out the previous gene expressed there, or through homology-directed repair, where a healthy DNA sequence is transplanted. While CRISPR is great because it's effective, its results are not always reliable.
With NHEJ, random indels can cause unwanted mutations, and with HDR, sometimes the cell rejects the transplanted DNA. Luckily, recent evolutions with CRISPR are allowing us to get closer and closer to getting the safer cure.
This occurs via a converter enzyme attached to a Cas9 nickase and a guide RNA. The gRNA locates the cut site. Then the converter enzyme changes the base pair, but now it doesn't match, so Cas9 nicks the unedited side, calling on the DNA to repair itself. By using a single compared to a double-stranded break, base editors decrease the opportunity for off-target mutations, but they are limited in what base conversions they can perform. Lastly, prime editing. The same Cas9 nickase and guide RNA are used, but there's also a pegRNA and a reverse transcriptase. First, the pegRNA locates the cut site, where Cas9 nicks the DNA. The pegRNA has two sections, allowing the reverse transcriptase to make a copy of the new DNA. But now the edited side doesn't match the unedited side, so the gRNA guides the nickase to cut the unedited side, so that then the DNA can repair itself. Prime editing has the pro of also using a single-strand break, but unlike base editing, it can edit almost any mutation. But prime editing, like the rest of the techniques, requires a lot more research to assure that only the safest tool is available.
As long as we keep researching and considering the ethical concerns, there is no doubt that gene editing will be the next best medicine to cure genetic diseases. Thank you for listening.